Okay, so Jung is going to tell us about his work on detection of coronavirus in Guinea bats. So the floor is yours.
Yeah, thanks for your introduction. And I'd like to thank the organizers and my supervisor for giving me a chance to be here and share with you my work in progress on Guinean bats coronavirus. Bats are the natural reservoirs of many important zoonotic viruses, such as the MERS or the SARS-1, SARS-2, and Ebola virus. And West Africa has been recognized as a susceptible area for zoonotic infectious diseases, as demonstrated by the outbreaks of Ebola virus and Marburg virus. And the current group at the University of Oxford is carrying out surveillance of viruses in bats in Guinea to understand the future risk of spillover. And for Guinea, the group focused mainly on the Gekadu and the Macenta prefectures, and has published a number of papers showing the evidence of circulation of Lassa filovirus, paramyxovirus, and coronaviruses. And the first Marburg confirmed case in the West Africa was reported from the Gekadu prefectures. So we carry out this study to understand if we can detect Marburg virus in these regions after we already obtained some serological evidence of spillover. And we also hope to detect some novel coronaviruses, specifically MERS and SARS-like coronavirus. And we'd like to understand their zoonotic potential as well. To do that, we collected the fecal samples of bats in Macenta in different villages, most of which the antibody against the Marburg virus were detected, that highlighted in red here. And sampling was performed by our colleagues Josephs and colleagues using the plastic sheets around the nest of bats. And so far, 480 bat fecal samples have been collected and processed. And analyses were extracted in Guinea and sent dry on columns to Oxford, where I eluted RNA and pooled 4 samples in one pool before DNA treatments, before CRISPR and Illumina sequencing. And the sequencing reads were classified using Kraken2 and also subjected to de novo assembly and then blast analysis. This is an overview of the NGS runs. Unfortunately, the viral reads accounts for quite a small proportion of the total reads in most pools, and most of the reads was the host from a host or the bacterial origin. We didn't detect any Marburg positive pools, and most of the identified viruses were insect viruses. This F1A tree shows you some, not all, but some identified coronavirus detected in Guinea bat samples, and they were highlighted in red. They cluster well with other 2290 coronavirus, another alpha coronavirus, bat nobecovirus, and another bat beta coronavirus. And this table summarizes the findings, including the closest virus, the number of positive pools, the sequence identity, and the contact length in KB. You see that beta coronavirus was commonly detected in the samples with the sequence identity just around 70% to the closest virus. So they represent novel coronaviruses there and potentially could represent novel species. Using PCR, I successfully amplified the full spike sequence of these 3 bat samples, and I cloned into the PCAC plasmid and co-transfected with other plasmids to produce the pseudoviruses. And I'm using the pseudovirus to understand the zoonotic potential of these viruses. with some advice from Dylan and Julia, I'm verifying the spike integration into the pseudovirus particle, and after that I will do the infection of different cell lines, including animals and humans. After that, I also perform the testing of receptor usage of these pseudoviruses. And because of the low percentage of the viral reach, I'm going to apply the capture probe method to improve the sequencing data outcome. In conclusion, coronavirus RNA was commonly detected in Guinean bats, and the obtained sequence show high genetic diversity and divergence from the known viruses. And the zoonotic potential of these viruses to be assessed using pseudoviruses. I'd like to thank all the members of the Carol Group. I'd like to thank Dylan Bailey and Julia Gallo and Pete Mays for the advices. The project was funded by the FDA and approved by the Guinean Ministry of Environment, Water and Forest. And thank you for listening.
Okay, so thank you for sharing your latest data. Do we have some questions? You need this one? Oh, okay. So I will start maybe. You show you got a lot of different samples, fecal samples. I was wondering if from those samples you could associate the virus with the species, like if you could tell us more about the representation of how many bat species have you tested, or is everything, I guess depends on the ecology of those bats, if they live together or not, but how many bat species were included in this sampling?
Yeah, thank you for the question. So you can see here that we just collected the fecal samples using the plastic sheet. Yeah. So it's really difficult to know how many bat species are there and which bat fecal samples we collect from which bat species. But what you can do is you can look for the mitochondrial DNA to see which species of the bat in there. Have
you done that?
Not
yet. Yeah, sorry. Okay. Yeah, we have some questions upstairs. Do you want to run? Who is it?
So, my question is, this was from 480 samples. Hopefully, you're going to have the opportunity to continue. What proportion do you think of the potential load of Betacoronaviruses sitting in those bat populations do you think you identified, and how many more trips are you going to need in order to be able to start to quantify that?
Thank you for your question. So far I presented. So far we obtained something like 480 samples, and we are still collecting more samples. And it's just fecal samples only, and we are going to collect more other samples as well. We are going to collect the samples at different seasons of the year and different locations to obtain more accurate the prevalence of the virus in the bat sample.
Hey, that was great. I was interested to see you detect some insect viruses in those fecal samples. Is that correct?
Yes.
What sort did the sort of the sort of abundance of those, or the different insect viruses, did they correlate at all with the ones that were positive for any of your coronaviruses?
For the insect virus, I found insect virus in most of the samples, and for some, it's at very high. It's accounted for quite a high proportion of the reach there. Yeah, so I think it's quite common to detect insect virus in the bat fecal samples. I don't know if the bat was fed with like insect or so.
Yeah, but I just wondered, was the sort of abundance uniform across all the fecal samples, or were there differences between different samples in terms of the sort of, I suppose, the virome of the insect viruses?
Yeah, so it's in different samples, but for some it's very, very high proportion, but for some it's quite low. So it depends on samples as well.
Cool, thanks.
Can you hear me now? Yeah. So We have a bit of time if there are more questions, otherwise I will ask something. It's a bit of a naive question. So my understanding is that Guinea is where we had the latest outbreak of Ebola. So I was wondering why you look for Marburg and not Ebola?
Yes, in fact, the primary outcome of primary focus of this project was the Marburg virus, because it's funded for doing study on Marburg virus, okay. And because this area were the Marburg virus was the first case of Marburg virus in West Africa was detected in Gekadu in 2021. So we hope we can detect some Marburg virus as well. And in fact, another group already studied bat viruses, and they found some Marburg virus in bats in Gekadu. Okay. We were a bit unlucky. We tried to look for virus in the placenta, but we haven't found one so far.
Yeah. Okay. Well, if it's all, we can go. We can move on from the next speaker. Thank you very much.
Thank you.
It was lovely.